Hello, everybody. I'm Tapa. I'm a postdoc at Princeton, and today I'll tell you about bacterial motility in porous media. This work has been done at Professor Sujit Datta's lab with Daniel and Jenna, both focusing on modeling bacterial chemotaxis. Bacteria are nothing new. We know about them for past 400 years, and we mainly study them in homogeneous materials. But in many physiologically relevant situations and in many environmentally relevant situations, bacteria live in complex porous environment. For example, they live in our gut mucus, in our intertissue pores, in soils and sediments. These are all complex porous environment. So if you want to model how infections spread through these tissues or how we can develop an effective bioremediation strategy, the first thing we need to know how these bacteria move through this complex porous environment. And currently, we understand this kind of systems by using a continuum scale reaction diffusion model where we treat bacteria as diffusive species. And two key parameters which are important in this process are bacterial diffusivity and flux of bacteria. But what sets those and how they depend on this kind of pore scale confinement, that remains a mystery. Mainly because most of the porous media that we can think of are opaque, and that precludes the direct visualization of bacterial motility inside porous media. And in this talk, I will tell you how we are solving this problem by developing a transparent porous media. We develop this transparent porous media by taking small hydrogel particles, swelling them in bacterial cell growth media, and we pack them really close to each other so they form this jammed packing of swollen hydrogel particles. Bacteria can live in between the interhydrogel pore space, and they can migrate inside this pore space. We can image this pore space by simply diffusing small tracer particles and taking a time projection of the tracer particle locations. And we find this pore space are really tortuous. And we can even track this individual tracer particle motion to measure the pore size distribution, and we find the pore sizes in this system are exponentially distributed, much similar to any other random porous media. And we can control this pore size distribution by simply changing the overall mass fraction of the hydrogel particles. Now we have a transparent porous media where we can visualize bacterial motility, and that's exactly what we do. And when you do this, we are completely blown away because we found that cells do not move by a run and tumble motion inside this material. Instead, these cells get stuck into really tight pore space. They constantly reorient their body. They eventually find a directed pathway where they hop to the next track. So they're hopping between tracks. So we found a new form of motility of bacteria inside porous medium. Now, what sets this hopping and trapping? Turns out hops are set by the pore space geometry, while trapping is set by a computation between pore scale confinement and cellular activity. So we can measure independently this hop lengths and the trap times, and from that we are proposing a new form of diffusivity that should emerge in porous medium. The diffusivity should be simply given by the hop length squared over trap time, because hops are when cells make most of the moves and traps are when they spend most of the time. Now, to verify this hypothesis, we inject a bolus of bacteria inside this porous medium and we measured their macroscopic diffusivity. We also predicted diffusivity from our measured hop length and measured trap time. Our measured diffusivity matches with our predicted diffusivity within a factor of 3, which confirms that by simply looking into single cell motility, we can predict the long-term diffusivity of bacteria inside porous media. But bacteria live as a population inside porous medium in many physiologically and environmentally relevant cases. But how would you possibly create a dense population of bacteria deep inside a porous medium at the right size, the right shape, and the right precision? But fortunately for us, our porous medium do have some self-healing material property. And during my doctoral work, I have developed a process of bioprinting any material inside this kind of deformable self-healing porous medium. And using this approach, we can directly inject a dense suspension of bacteria deep inside porous medium, and we can make any structure out of this material at any kind of given size, shape, and aspect ratio. To start with, we make a small cylindrical structure of bacteria. This is a cylinder of bacteria which is embedded inside a porous medium. Each individual pixel here represents one single bacterium, and you're looking into this structure from top down. This overall field of view represents thousands of cell lengths. When you watch this structure over time, we're completely blown away because the cells start to form this beautiful traveling waves. Let me remind you, each individual pixel here is one bacterium. Thousands of cells are moving collectively throughout this pore space over thousands of length scale. So what's happening here? Since this process happens over about like 14 hours, you might think that these cells are barely growing and pushing their progeny forward. They are not moving. But that is not true. If you zoom in here in one of those traveling waves, you'll see that cells are motile. They're moving at random direction, but the overall front moves at a constant speed at a constant direction. And since we are collecting this data on a confocal microscope, we can look into this process from the side and we can verify this is truly a one-dimensional problem, where a solid cylinder of bacteria is expanding as a hollow cylindrical shell. And we can collect this data and we can azimuthally average the fluorescent intensity and plot them in a single plot to question how fast these traveling waves are moving. And we repeat this process at 3 different pore sizes, and we find that as we decrease the pore size or the pore scale confinement, the traveling wave speed decreases. Now, what sets these propagating waves? Bacteria are much like humans. We go to a new place, we colonize that place, we deplete that place out of its local resources, and eventually we want to go to another place. Bacteria are nothing different. As soon as create this green population of bacteria, which is embedded inside this porous medium filled with orange nutrients, they start to consume their nutrients around their vicinity, eventually set up a nutrient gradient, and they start to chemotax towards their own nutrient gradient. Since there is a clear separation between the length and time scale between individual cell motion and the motion of the propagating wave, we can use a continuum scale reaction diffusion model to describe our system. And in this model, the concentration of nutrient at any given time is given by nutrient density and nutrient consumption, while bacterial density at any given time is given by bacterial diffusion, bacterial chemotaxis, and their net growth. Now, the bacterial diffusivity and this chemotactic parameter, chi, are usually taken as an ad hoc parameter in most of the modeling. But since we can measure the bacterial diffusivity directly inside porous medium, we can implement that diffusivity in our model and question if this chemotactic parameter is really a bacterial intrinsic property or it depends on the pore scale confinement. And our reaction diffusion model qualitatively predicts this kind of traveling wave formation. And we can repeat this process at 3 different pore sizes to find that not only our model predicts this traveling wave formation in porous medium, but it also confirms that these traveling waves actually follow the nutrient gradient. So bacteria are actually following their own nutrient gradient. And since we are measuring this diffusivity directly inside porous medium and implementing this diffusivity, we find that chemotactic parameter is also a pore scale dependent parameter. It doesn't only depend on bacterial properties. Now, how the seemingly random movements of bacteria generate a propagating front? In more general words, how bacteria can chemotax in porous media. And surely we did not discover chemotaxis. Chemotaxis is well known for years, and in liquid culture, we know that E. coli cells can chemotax towards a more nutrient-rich zone by simply elongating their runs towards the more nutrient-rich zone. Now the question is, can they do the same thing? Can they hop for a longer period of time towards the more nutrient-rich zone? Our hypothesis is no, because that is not possible. As hops are set by pore space geometry, cells cannot move farther than the space available to them. Can they truncate their hops in the opposite direction? Turns out that is also not possible because pore-scale confinement suppress flagellar unbundling, so cells cannot truncate their hops midway through. And to verify this, what we have done is we tracked hundreds of cells at the leading edge of the propagating wave, and we measured their hop length, and we found that there is no directional bias of the hop lengths. So how these cells are even chemotaxis? Turns out there is another mechanism by which E. coli cells can bias their motion. They can move towards the more nutrient-rich zone by biasing their reorientation amplitude. In simple words, when they tumble, they move more towards the nutrient-rich zone. And E. coli cells can actually do that. They can bias their reorientation amplitude by involving different numbers of flagella into the unbundling process. We think since cells cannot elongate their hops in porous media, this secondary mechanism is probably the dominant mechanism of chemotaxis in porous media. And to verify this, we measured the probability of hopping at different direction, and we find that indeed cells are more probable to hop towards a more nutrient-rich zone. And finally, to verify the relative contribution of these two different mechanisms, we calculate the chemotactic front speed from the hopping length and trapping time at different directions. In this equation, if we replace the hop length, the directionally dependent hop length, by an average value, that does not change the overall front speed. However, if we change the probability of hopping or the directional dependence of hopping by a uniform distribution, that completely changes the chemotactic front speed that we measure, which confirms that this anisotropic hopping is the dominant mechanism of chemotaxis in porous medium. So we have found that a new form of motility emerges in porous medium, and this new hopping trapping motility is determined by an interplay between pore-scale confinement and cellular activity. We can independently measure the hop lengths and trap durations from which we can calculate their long-term diffusivity. We found that confinement regulates but does not suppress collective migration, which happens via chemotaxis, and chemotaxis is controlled by a secondary mechanism of anisotropic orientation that happens in porous medium. So far, what I have told you today is either about uniform distribution of bacteria, where we study their diffusivity, or homogeneous population of bacteria, where we study their chemotactic parameters. But what if your bacterial population have different shapes? To answer that question, we create this sinusoidal population of bacteria, which is embedded inside porous medium, and we watch this over time. And we see that bacterial chemotaxis towards their own nutrient gradient smooths out any morphological perturbation that happens in this system. We use a continuum scale reaction diffusion model to verify that chemotaxis is the main driving force in this morphological smoothing process. So what sets this chemotactic smoothing? Turns out cells are most chemotactic when they're sensing a concentration of nutrient within this window of concentration. And in this simulation, we have indicated this upper bound and the lower bound of the sensing window by these two different red lines. And as you can see, at the convex region, the nutrient concentrations are way above this upper limit of sensing window, which means cells are less chemotactic at this convex region. They move slowly, which controls this chemotactic smoothing. I have told you today that how we are combining soft matter engineering, cellular biophysics, and active matter transport to make new discoveries in bioengineering. Now we have developed these tools and techniques. I want to use this as an independent researcher to create designer cellular communities and 3D biointerfaces. I want to make designer multicellular communities of controlled size, shape, and spatial composition. I want to probe them at complex biological interfaces. For example, I want to study how cells self-assemble at curved deformable interface. I want to make microtissues and probe them for different mechanical stimuli. I want to study how bacteria can evolve and adapt to a competing chemical gradient. I would love to talk to you more about these ideas that I have. Please feel free to reach out to me, and I would like to take any questions that you have. And with that, I would like to say thank you, and I'm looking forward to your questions. Thank you.